I'm very excited today to talk about my latest results on the infant gut microbiome and mother's breast milk. So we all know that the infant gut microbiome plays a very important role in maintaining and defining health in early life. But also we know that the first weeks and the first months of life are really important because during this time window, the infant gut microbiome composition can really be influenced by a variety of factors, among which the influx of maternal microbes. So over the years, there have been several studies that looked at the impact of microbes from different maternal body sites. But one aspect that has been consistently overlooked is breast milk. So breast milk is very important for infant health because it represents, in an ideal situation, the only source of nutrition for the first 6 months of life. And it does also contain, among many other things, live bacteria that have a direct potential for impacting the composition and the assembly of the infant gut microbiome. And also milk, despite all of this importance for the infant health, milk remains heavily understudied. It represents less than 0.5% of all the human metagenomic samples that are available on public repositories. So with this kind of context in mind, we set out to basically address two questions. First, can we improve the representation of breast milk in public metagenomic repositories? And can we find evidence of strain sharing between breast milk and the infant gut? So to address these questions, we reached out to our collaborators at the University of Minnesota, and we looked at a cohort of 195 mother-infant pairs for a total of 507 samples. We looked at breast milk composition, breast milk microbiome composition at 1 and 3 months postpartum, as well as the infant gut microbiome at 1 and 6 months of age. As we were expecting, we found that maternal breast milk microbiome diversity was significantly lower than what we could find in the infant gut. But also when we looked at this ordination plot here on the right, you can see that the composition of infant stool samples is very different than the one of breast milk samples overall. However, when you look actually at the composition in terms of heat maps of the breast milk here on the left and the infant stool here in the center, side by side, you can see that actually Bifidobacterium longum is driving the overlap between these two body sites. So B. longum was actually the most prevalent and the most abundant species that was found both in breast milk and in the infant stool samples. And also we were able to dive a little bit deeper into B. longum, and we found that in both milk and infant stool samples, the two main subspecies of B. longum infantis, B. longum longum, and B. longum infantis were actually present together, were coexisting in the same samples in the vast majority of cases. But as I said before, we were very interested in looking at microbial taxa that were shared between breast milk and the infant gut. We found that, for example, Bifidobacterium longum infantis was among the species, or in this case, subspecies, that was most commonly shared between mothers and their infants, was found in 47% of our mother-infant pairs, followed by B. longum, S. epidermidis, breve, and salivarius. At the strain level, which is the part that we got most excited about, was actually that we found several instances of strain sharing events between the maternal breast milk and the infant gut. These are just some of the few phylogenetic trees of the taxa for which we found these instances. The strain sharing events are highlighted in the black square. And for example, for B. longum, in this case here on the most left, you can see that we found a specific strain of B. longum in the mother's milk at 1 month. We also found it in the infant gut at 1 month, and that same strain was able to persist in the infant gut until 6 months of age. So overall, we found strains that were shared between breast milk and the infant gut that included not only commensal taxa like Bifidobacteria, but also pathobionts like Klebsiella pneumoniae. We also saw that the percentage of taxa that were shared between breast milk and the infant stool decreased over time. So it was higher at 1 month compared to 6 months. And on average, 10% of the taxa found in the infant gut at 1 month were also found in the mother's breast milk. We also took a step further, and we didn't look just at the microbiome composition. We also looked at antimicrobial resistant genes. And we found that basically milk samples, both at 1 month and 3 months, were characterized by an extensive carriage of antimicrobial resistant genes, mostly belonging to the classes of tetracycline, beta-lactam, and MLS. And there was also significant variability in terms of the percentage of antimicrobial resistant genes that were shared between the mother and the infant across different pairs. So some pairs had very little percentage of sharing, some other pairs had a much higher one. And overall, mother-infant pairs shared a significantly higher number of antimicrobial resistant genes than what we would expect by chance. And finally, we also found that infants whose gut microbiome was dominated by Bifidobacteria were characterized by a significantly lower carriage of antimicrobial resistant genes than the infants whose gut microbiome was instead dominated by other taxa. So this is just a very quick summary of the main findings that, of our study, but you can find more results and more interesting discussions about this interesting results on the publication that was recently got out in Nature Communications. And before finishing, I also would like to thank my colleagues at the University of Chicago in the Blackman Lab, as well as our collaborators at the University of Minnesota, and most importantly, all the mothers that really volunteered for this study and made this analysis possible in the first place. Thank you so much.